So, very good evening, all of you, and welcome to the first online session for the course Pharmacognosy and Phytochemistry. So, I'm Galvina Pereira, your instructor for this course, and I welcome you to this first Q&A sessions. We've been receiving a lot of support as well as questions on the forum, and till then, I would request you to share your questions in the chat boxes. Meanwhile, I'll just discuss a few that were pertaining to this course. So many of you have inquired about the course content, especially the study material. So the study material for this course could be any of the textbooks on pharmacognosy and phytochemistry. So all the textbooks in pharmacognosy and phytochemistry do contain the content of this course, that is the phytoconstituents as well as their quality control methods. So some of you specifically asked for books. So we can start from Trees and Evans, Pharmacognosy. We can have your Pharmacognosy by Wallace. Brunitin, we have Pharmacognosy by Gokhale, Kokate, and Purohit. Um, Many other books are also available. So all of these books which discuss the drugs in detail can very much be the course material for this subject. So do let me know if you're finding difficulty in analyzing or assessing content from these books. Now, because this course is a relatively short-term course. Now, when we were preparing this course, we were preparing taking into account semester 5 contents as well as some of the semester 6 contents. But owing to the time restrictions, if you observe, we don't go, or our team, we've discussed, and in terms of its syllabus, I'm not dwelling much into the cultivation, collection, and little microscopic part of the drug. In some cases, we've kind of abbreviated even on the chemical test as well as general cognosy section. So this is just keeping in mind that the course is going to last for 30 hours. But yes, we would really want you to go through this sections also when you see the textbook or when you see the notes. The text transcripts for this course will be uploaded and will be available shortly for you. Now, coming to few questions that were raised in terms of the forum, apart from the notes, yes, we have a question on maybe crystallization of honey. Maybe there was a question saying that what happens when your sugar crystallizes in honey, whether it's a genuine honey or not. So taking into account that, what happens here is we saw that in honey, when you talk about the specific gravity or when you talk about the density of it, it's very viscous. The sugar content may be as high as 75% in your raw honey. And that is the reason the tendency of raw honey to crystallize is much, much higher than the processed honey, where the specific gravity is brought down for ease of manufacturing, processing, as well as for consistency. But yes, in some cases, when you say it is adulterated deliberately by adding more and more sugar, if your sugar content goes equally high, even on adulteration, more than 70%, then there is a high chance that you'll observe a crystallization therein also. This was a case observed with one of the companies, Himalaya, and you would seen some of their products depositing a little bit of crystals. So yes, in some cases, genuine honey as well as adulterated honey, you will find crystals. But adulterated honey, you'll find crystals only when the content goes very high, as much as 75% of the sugar content. And on evaporation or on keeping, the water evaporates. Or even on cooling, when you keep it in fridge, when the kind of water tries to decrease the solubility of it, the water can no longer hold those sugars, and as a result, it precipitates or crystallizes. Now, another question was in terms of solubility of gums. Gums, as such, most of them debate whether they are soluble or not. In terms of Indian pharmacopoeia, even we check acacia, it is said that whenever you have a gum and it is swelling, it is to be dispersed. You either take a mortar or pestle or you take a glass rod, and when you dilute it sufficiently, it is or it does achieve a consistency where it can be filtered, especially through your G2, G3 sintered glass furnace. In that case or in that context, it is called as your soluble, or they're called as water soluble. So that they also compromise water, or they also comprise of what are called as your water soluble fibers, which make up your prebiotics. Any queries apart from that can be posted in the comment section. Now you can also post in your feedback or specific regards to the quiz or any question you found difficult encountering or tackling while you were solving the quiz. Meanwhile, we'll just see a few questions which are there on the forum. I think on the forum, majority of the queries are pertaining to the notes. So we'll upload the text transcripts of this shortly. There's a question pertaining to mineral-based drugs such as Shilajit. What exactly is fossilization? So when we are talking fossilization, basically in terms of Shilajit, what happens is there are numerous microorganisms, or there are numerous fungus or algal species that exist. So in our context, let's take for example certain algae or fungi. Now, this certain algae or fungi that are growing during the course of time, imagine they get somewhere submerged, or they get kind of uh, buried is a more proper word, into the crevices by the subsequent soil erosion which happens and which covers them entirely. Keeping that in mind, imagine a process which keeps on happening and your fungus, algae are still sustaining under the soil. What happens is gradually they'll start a process of decomposition. So they'll become brown, then they'll become oleaginous, and then they'll become very carbonaceous. So that is what we refer to the process of fossilization. So they become rich and more of your organic content, and slowly start getting converted into crude slicky substance, and this crude slicky substance still retains certain metabolites. So in case of Shilajit, it still retains the fulvic acid. So despite having undergoing or starting the process of fossilization, those components are very active and help in the process. So, yeah, there's a question from Harish asking, what is the role of flavonoids in different colors of plants? Now, the colors of plants, let me divide it. So when we say colors of plants, the green color is of the leaf is generally attributed to chlorophyll, and flavonoids have no role to play there. Now, taking into account few drugs which are colored, especially your take for an example your kokum, or take for example certain flower petals such as rose. In that case, you come across a flavonoid category called as anthocyanins. So anthocyanins are colored pigments, and depending upon their substitution, you can have a really red coloration like a cherry, to purplish coloration like a petunia, to almost a dark coloration of concord grapes. So all of this can be brought about by anthocyanins. So from orange to pink to purple to almost deep blue in coloration is something what flavonoids can bring in. Now, if you think of class apart from anthocyanins, flavonoids are off-white to pale yellow in color. So you might see certain flavonoids such as rutin, quercetin. In their purified form, they'll be yellow to yellow greenish in coloration. Now, the role that the flavonoids play is when it is anthocyanins, the important part is to attract insects for pollination. And in some cases, when you say your flavonoids are a part of leaf surface or a stem surface, definitely the action there is more of free radical scavenging or a sunscreen kind of effect. Oh, there's a question from Alexander that can anticholinergic agents such as extracts of atropa be made into wash cream or to reduce Palmer hyperhidrosis? Now, the mechanism of it is atropine should first bind to receptors. Now, we need to find out on the surface of hands, do we really have such kind of receptors? So, if you have what you call it as, cholinergic receptors, which I'm not sure at this point there exist. We need to find on that. In that case, only it would be possible. Otherwise, you'll have to take it internally to kind of prevent sweating. Unless and until you have those receptors, especially your cholinergic receptors on surface, the mechanism of it won't be working. So I feel at this moment, since I'm not aware of the receptors on the surface of hand, I feel taking it internally would be more advisable. So, do you have any resources or suggestions for learners who don't have a background in chemistry? Okay, so in Cognosy, we do address little bit of chemistry, but in terms of resources, uh, preliminary reactions, what we study, or certain analytical books are something that I would recommend. Analytical books like pharmaceutical analysis, or you can check for Beckett and Stenlake for analysis. Those kind of preliminary books will help you out. There's no in-depth. What we are more interested is to check the reaction of phytoconstituents, to give you chemical tests and analysis, and maybe the chemistry, how it is related to pharmacology. So that is more elaborated in medicinal chemistry books. So you can check maybe Wilson and Giswold or Foe. But if you're looking more for a chemistry which is relevant in terms of applications, I feel, or analysis, I feel Beckett or even simple reactions would be sufficient for you. So Beckett and Steinlake for analytical part of it. Foe if you want to understand a little bit of medicinal chemistry. And majority of the discussions in pharmacognosy, especially in this course, happens in analysis. So any book on pharmaceutical analysis of phytoconstituents would also help you out. We just get back to the questions on forum till then. Some of you have queries about the certificate. It is stated on the course outline. What happens here in the NPTEL is they have a examination which happens at the end of course. You exam register yourself and complete the exam. So you have a kind of distribution where you have almost 30% marks for internal assessments as well as 70% marks for your end examination. So if you qualify that, in that case, you are eligible for certification. Then the query is how do you get a PPT or PowerPoint presentation notes? We'll be providing you the text transcripts which also contains the screenshot of the PPT as well as the spoken content during the session. So this will be uploaded shortly. It's currently in process of approval and uploading. So that's happening simultaneously. Once it is released, we'll be broadcasting it on the same course so it will be accessible to you all. There's a query on solvent of extraction. So just address it. How do you select solvent? In this case, it is for volatile oil extraction, but we can discuss it for most of the compounds. Now, what happens here in case of volatile oil is generally, we go by formula like dissolves like. So whenever I have to extract anything polar, I will choose a polar solvent. So for example, tannins, which are polar, you can get your tea, which is rich in tannins, or coffee, which is rich in tannins. So in cases where there are drugs containing polar compounds or drugs containing non-polar compounds, but in the glycosidal form, because the sugar is attached to them, that makes them polar. So essentially, you can start with a very simple solvent for their extraction, that is water. Saponins are extracted in water. Your glycosides, majority of them are water soluble. And in some cases, your tannins also are water soluble. Now, in case where you see a bigger nucleus, but lot of polar groups. Now, these polar groups include your hydroxy groups, amine groups, or any other charge moieties which are attached on a very hydrocarbon nucleus. We saw it in carotenes also, be it even C20 or C40. Once the sugar or an hydroxy is attached to them, they come into the category of mid-polar compounds. So for mid-polar compounds, generally solvents such as ethanol, ethyl acetate, isopropyl alcohol, butanol are chosen. Now, coming to the lipophilic category, imagine molecules something like your steroids or oils or volatile oils. In that case, a good solvent for extraction would be a lipophilic solvent. This include your hexane, benzene, toluene, petroleum ether, diethyl ether, and so on. So you can do that. But then the question is, why is water there in your volatile oil extraction? Now, this is based on a phenomenon, what we call it as steam distillation. So here, the only reason water is used for extraction is water, when it is boiled, it kind of volatilizes the essences of the drug. That means all the essential components of that drug would become volatile. Now, attach it to a condenser. What is going to happen is all this oil which has volatilized is going to condense, and so is your water. So in the process, your water will also condense and your oil is also condensed. Now there is a little bit of lipophilicity difference between the two. At the same time, a majority of the cases, there is also a difference of what is called as specific gravity. Some oils are lighter in water, some oils are heavier in water. Very rarely you will find oils with a very same density or specific gravity as that of water. So you will have either your oil settling down or you will have your oil floating up. That makes separation much easier than extracting it in a non-polar solvent. Say for example, if you extract it in chloroform or toluene, what is going to happen is your chlorophyll and your carotenes and other pigments which are non-volatile are also going to come in. So in this case, water, because it is a heat-based extraction method, becomes more selective, and that's the reason it has been chosen for your volatile oil. We just see few more questions on the portal. So there's a question about, does degumming require heating or not? Now it is entirely dependent on the drug, but in majority of the cases, degumming uses heat. So degumming is a process for refining of your fixed oils. Imagine most of your cooking oils, when you kind of express them, what is going to happen is your oil, when it is coming out, other plant juices also come out because there's a high amount of pressure being involved. So in this pressure, it's not only oil which is coming out, it is some of the compounds which are oil soluble also coming out. It is the juices of the plant which is also coming out, including those located in seeds. Now, certain times what happens is there are gums or mucilaginous or more of carbohydrate rich matter which also comes down. Now, what is going to happen is this is going to create an emulsion like layer, because it's gum, and you know gums create emulsion in presence of water and oil. So in that case, especially if the oil content is good, in that case it is more advisable to heat it, because heating will crack the emulsion. And as soon as it cracks the emulsion, you will get a good separation of water phase and oil phase, and your gums, being carbohydrates, will be restricted or will be more seen in the aqueous layer as compared to your lipophilic layer. So heat is definitely preferred, but there are certain cases in which the water content is higher and the oil content is lower. So in that case, you need to take a judicious call that maybe your process doesn't emulsify it more, or if you're heating it, you heated it to the such an extent that all the water is evaporated and you're left with only oil. So as the water goes on evaporating, what will happen is all this gum will eventually dry up as a film on your vessel, and your oil will kind of separate out. The same way we cook onion or tomatoes, you might have seen. They are carbohydrate rich, but as they kind of segregate, you will see the oil splitting out. So that can be done if the oil content is less and you can evaporate more of water to get away. Okay. There is a question regarding formulating quality control standards for ethnomedical plants. So, Mr. Swapnil, yes, I do take that, and there is a huge need for it. The reason being, we are a country predominantly where still we prefer kadhas, where we prefer adulsa syrup, where we prefer a lot of herbal material. Not only that, we as a country have a rich heritage even with folklore medicine. So even if you go to villages, there are n number of plants of which we might not have studied in our curriculum, but the tribals or the local or the native people out there very use it very frequently. So in that case, what we generally suggest or what can generally be done is you to pick up a few quantum, like few kg or a specimen from different regions, and try to find out using phytochemical analysis. We'll be coming short to that in your week 10 and 11. So you can do your phytochemical analysis, you can standardize it, what all it contains. You could do a maybe little bit of TLC and start creating a monograph. So when you see your Indian pharmacopoeia currently, you have a whole section of monograph. So maybe you can take this initiative and prepare or start preparing a guideline for that particular plant in the same lines and maybe publish it So tomorrow, anybody who's handling the herb can have your publication to the reference of it. So I think, Swapnil, if that answers your question. Now, what is the procedure for isolation of clove oil? Now, clove oil, Harish, is actually a oil which is denser than that of water. But when I say density, it is water is 1, clove oil is 1.08. It is not 1.8, it's 1.08. So it has a density very, very close to that of water. So when you're doing clove steam distillation, especially, many of you who have performed this experiment will know that it doesn't initially come out as a separate oil and liquid. It comes out as an emulsion, and this emulsion takes a sufficient time to kind of separate out. So we can still facilitate it by first thing is your condenser. You cool it more, probably circulate a very chilled water. In that case, what happens is those clouds, tiny droplets, they will condense quickly, mutually coalesce, and once they coalesce, they form a larger droplets. And larger droplets, it is easier for them to separate it later. Now, if you already collected the emulsion, one thing what we can do is generally either microwave it. Sometimes even microwave creates a good density difference. Just microwave for 10-20 seconds and see what happens. So maybe you're heating, but be careful, don't overheat it, otherwise the clove oil will volatilize. So just keep the cycle short and see that the clarity is achieved by, because on heating also this viscosity decreases and the droplets coalesce. At the same time, another method is when you have emulsion, you could also do a salting out. And this increase in salt can also help you segregate the clove oil. So, clove oil is generally purified by steam distillation, and the distillate is collected, allowed to stand for a while till the emulsion separates into two distinct layers. If the emulsion still persists, you can increase the temperature or give it short, sharp cycles of microwave if you're collecting it on a smaller scale. If you're collecting on a larger scale, like in a liter scale where that is not possible and you still have a lot of emulsion with you, what you can do is you can take help of certain solvents like your dichloromethane. Dichloromethane will kind of dissolve, like dissolves like principle, all the clove oil, and later on you can separate the DCM layer and carefully evaporate it. Now, on careful evaporation, the DCM will go and your clove oil is left back. Or you can even do it with other organic solvents which are chlorine free. So there's a question from Anushka asking that whether, like your lignans are a type of flavonoids or they are a different class. Now, what happens is the week in which we have flavonoids, I guess that is the week where we have all the different classes belonging to phenyl propenoids. So just to put this correction, that is the week on phenyl propenoids, and in phenyl propenoids you have flavonoids, you have lignans, and you have coumarins. All three of them belong to different classes. Flavonoids are more of what are called as phenyl benzogamma pyrrole nuclease. Whereas if you want to see lignans, lignans are nothing but they are dimers of phenyl propenoids. So they are structurally very divergent. Coumarins are mono. They are just phenyl propenoid. So all of three of them are phenyl propenoid derivatives, and that week is particularly, I think it's week 8. It's more about your phenylpropenoid derivatives, and flavonoids, your lignans and coumarins are three different classes. So we have question from Alexander. What is the best and safe way to extract lotus heart, a small green, bitter inner part of lotus seed for home use, for assisting in sleep and relaxation. So, Alexander, that's an interesting question. What happens is when you see a lotus seed, now lotus seed will be having different set of compounds. Now, let's say at this point of time, we are unaware of what it is. Maybe at your end, you would have done little bit of research. For me, this is a new plant, so this is how I would tackle it. Now, if it is a new plant to me, I definitely, depending upon your question, have one idea that it contains something which are bitter in nature. And the second thing, what I get to know in terms of its application is somewhere this is going to assist me in my sleep disorders. Okay, so for this, we call what is called as bioassay guided fractionation. So, what is this bioassay guided fractionation? It is very simple. You take that lotus seed heart, or what we call it as the endosperm or cotyledon, or what specific part you want to take it. Okay, that part you see whether it is, reported to have or possess certain constituents or not. That means, have the phytochemical studies of these plants been done earlier, or are they entirely new? Now, let's say we go with the first example. I know what the phytoconstituents are. Now, if I know what the phytoconstituents are, what those bitters are, or what those sleep-inducing compounds are. So, say for example, those sleep-inducing compounds are steroids. I know it very clearly. In that case, my work is half done because I know it is steroids. It is going to be lipophilic. So, the moment I extract my lotus seed with anything lipophilic, it is going to pull out those compounds. Now, coming to the case B, where I don't know what it is. In that case, bioactivity guided fractionation will tell you that you start extracting the seed with a grade or with a subsequent extracts. Now, this subsequent extraction should be done with more and more polar solvent. Say, for example, the first thing you will extract your lotus seed is something like your petroleum ether or something like your toluene, something extremely lipophilic. So all my fats, all my triglycerides, all my lipophilic substances will go. Now you know seeds are very much rich in lipophilic substances. So once this lipophilic substances go, now your seed or seed powder or your seed heart powder is more pervious to the polar solvent. Otherwise, the water will not go inside. You need to take that lipophilic coating off, and that is done by defatting. And defatting is done by organic solvent. Now, once the defatting is done, you can start with the subsequent solvent. The subsequent solvent should be little less non-polar than the previous one. So if I've chosen something like your petroleum ether or toluene, the next little polar but still non-polar would be something like your chloroform, dichloromethane, or acetone. You can extract it with that, and then subsequently a little still more polar would be alcohol, and most polar will be water. Now, say for example, I have this four extracts, say petroleum extract, chloroform extract, alcohol extract, and water extract. Of these four extracts, I will do my activity studies, and I will check which of this extract is really containing compounds which are sleep inducing or sleep causes relaxation. In this case, suppose I get a chloroform extract, which is going to be more active. Then I will do maybe column chromatography or certain purification techniques, and then try to purify and fractionate all the compounds which are present in the chloroform extract. Or alternatively, nowadays we have what is called as plant metabolomic studies. So you can give your plant to a LCMS or HRMS studies, and then they will characterize using certain softwares like Melton to quantify what all compounds are present in that extract. Based on that, you can make a conclusion that these are the compounds which are responsible for the sleep inducing effect or sleep assisting effect of the lotus heart. So Alexander, just to be clear, so you used a decoction, so you used alcohol in that. So in that case, when your alcohol is giving you more of this or if you have this drug in a more quantity, you can still carry out phytochemical evaluation of the alcoholic extract and check for compounds such as tannins, flavonoids, and other derivatives. You can still do a small preliminary evaluation and then subject it to plant metabolomic studies. You'll get more of the details once you do a HRMS or a plant metabolomic studies. But if that is not possible, a very simple thing to do is just carry out a preliminary phytochemical evaluation, do a total polyphenol content, do a total and like a flavonoid content, and you understand what all compounds it contains. And then you can make a comment, probably this set of compounds are responsible for the activity of the lotus heart seed. So I'm just seeing few questions. Normally in East Asia, it's made into tea. Okay, so there's already a report that it is used in form of a tea. And when I understand it's a tea, definitely it's going to be a water extract, right? So if it is a water extract, there's a good chance that there are tannins in it. There are flavonoid or especially glycosidal compound in it, which are extensively making it polar. So try to understand which all glycosides are present or are reported in that. If they're not reported, maybe you can do a little quantification assays to check which all sets of compounds are present. So do a preliminary phytochemical assessment of that tea, followed by probably a TLC if possible at your end, and then subsequently you can go for a characterization. Definitely, I'll wait for that. So once you do a research on it, do let us know. We'll be happy to help it out. Somebody is inquiring about the answer key to quiz 0. I think it's already there. Yes, majority are still about the notes. So the transcripts are on the way, and maybe you'll find them uploaded in the short while. Meanwhile, any feedback on the course or on the quiz contents, whether they are understandable, tough, or relatively easy, feel free to suggest it so that like we can get a good feedback for developing the course content subsequently. Swapnil, one more thing is probably in your week 11 and week 10, we'll have more of this quality control methods. So maybe, we can also discuss something about your ethnomedicinal plants there. And if not, what we can do is maybe, next time in our forum, we'll bring it. Maybe you can share the plant you want to discuss, and we can just take a case study of that plant and discuss what can be done over it. Meanwhile, just to elaborate, when we have our compounds, especially the flowers of it, what you see is there are certain flowers which are anthocyanins or flavonoid containing, but yes, there are certain flowers like marigold which you might come across. So there, what you will find is the pigment is not flavonoid. The pigment may be carotenes. The pigment may be betannins. So, say for example, your beetroot or bougainvillea. The bougainvillea flowers are due to betannins, and the flavonoid pigments are absent in this plant. Any more queries or questions you have pertaining to this course? Yes, there was a query on assignment of week 3. I think there was a spelling error in iota, and that's the reason majority of them or some of them got a question error. So we've resolved that query, and it's no longer the question is considered. So your quiz will be considered devoid of that particular question. Just checking on the forum. Okay, Swapnil, so the query is what is the importance of powder microscopy? So, if you're talking about the relevance or importance of powder microscopy in the subject of pharmacognosy and phytochemistry, it's really, really relevant. So, the first thing what we do is we definitely see the gross morphology as well as macroscopy. But if you see the real life scenario, what happens is majority of the times when your drug material comes to the industry, you get it in form of powder. And the reason is very simple. If you get it in form of crude drug, again there are chances of insect infestation. Imagine your entire leaves or wood being there or roots being there. So there's a good chance of insect infestation. Not only that, it increases a unit process for that industry. That means that industry has to have an additional room where the work would be just to take this raw materials and convert it into powder. So many of the industries prefer getting the raw material in form of powder. So as a result, it is good to understand the quality control of the drugs from the powder perspective. So what do we do in that case is when you see powder uh, microscopy, we are actually seeing certain features. Maybe these features can be botanical features like your xylem, phloem, cork cells, or which are more relevant to that plant. And in some cases, this powder microscopy will expose me if that supplier is kind of providing some adulterated substances. So imagine just seeing a xylem in a bark. You know, your bark is something which is separated from the cambium, and at no point it is bound to contain a xylem. But if at all a xylem has entered in, that means somewhere your stem was also adulterated there. So, giving this example, not only that we've seen stone cells, sometimes we see covering trichomes, those representing the leaves being seen in bark, being seen in root. So that gives a good indication. Even when we see them under microscope, we know that that is adulterated or that has been not collected or harvested properly. Some part of leaves, some part of stem, some part of roots have been deliberately or indeliberately come into your drug, and as a result, once that was powdered, everything came into that. So, an indication of any foreign substance into your drug, especially powder, is an indicative of an adulteration. So, is there any other assignment, homework, etc. planned which would give more experience, practice with the course material? So, the assignments, homeworks for you are the assignments that happens especially after the end of every week. So that's the just a small quiz to see if you understood it or not. In addition to that, we're not having any assignments for this course. But yes, we do request or expect you to go through the textbooks for this drug materials. Why? Because we've abbreviated this course to fit into 30 hours. So we definitely really appreciate if you take your time and interest into reading the textbooks in pharmacognosy, which will give you much more depth about the drug, even more than what is discussed in this course. Then there's a question from Harish. Can you differentiate between primary and secondary metabolites? So, primary and secondary metabolites is what I say is when I want to survive, anything that will help me survive, that is, provide me with food, provide me for my growth. All those substances which are very vital for my growth as a plant, I put it as primary metabolites. Now, apart from that, there are some metabolites. Say, for example, the color of flowers. In some cases, you will see that even if there's no color to flowers, I mean, that doesn't really stop the plant from existing. So, in that case, or there is no aroma to the flower, there definitely it has pollination. The trichomes have defensive function. Those bitter compounds will prevent that plant from being consumed by animals. So these are all secondary functions. Your plant will not die without it, but that increases the plant's chances of survival. Such metabolites, I put it as secondary metabolites. So primary metabolites are more often the molecules which give structure, which help plant in growth, whereas secondary metabolite are the ones which help in maybe reproduction, attraction of insects, or even defense function, help it to kind of sustain in drought, help it to kind of sustain in extensive rain. All those metabolites, I'll put it as secondary metabolites. So there's a question if you have to do a transverse section analysis versus powder analysis, which is more useful or understandable. Now, In terms of understandable, the my answer, or this is my personal answer, I feel a transverse section is definitely more understandable because a transverse section is like a complete painting, whereas your powder is a jigsaw puzzle. So when you see a powder, you have to kind of put it. This is a xylem, probably this would be here. This is the cork, probably this would be here. So it is a part and parcel of your transverse section but converted into a jigsaw puzzle. But when I say a transverse section, it is a whole painting which is made nice and clear to me. So, in terms of understandable, definitely it would be a transverse section. Powder, I would call it as the same painting. If I cut it into tiny, tiny pieces and give it to you, I call it as powder microscopy. But understanding is a different thing. What is more useful? Now, in terms of its utility, like I said, majority of the vendors or majority of the industries purchase your raw material as powders. Now, powders definitely you could do powder microscopy. Powders you could definitely do your chemical test. So, powder will not make you confident that this is definitely a particular drug because in some cases you might be able to solve that jigsaw puzzle, in some cases you might not be able to solve that jigsaw puzzle. But the knowledge of powder microscopy is useful when it comes to its application in industry. So definitely when you blended three powders, say harda, bheda, amla in my trifala churna, I should be able to see the powder features of all three. If I'm not able to see, I'm just able to see the powder features of two drugs, that means my jigsaw puzzle is incomplete. So that's the little difference between your transverse section and powder. Both have its own relevance. But in terms of understanding, again, I still go with the transverse section. It's much, much simpler and easier to understand. Now, any powders that you have analyzed and come across a difficulty understanding where or how these powders I'll understand, do let me know. Maybe we can have a little trick to that. So the trick to that is first understand the plant part. And once you understand the plant part, maybe stomata or some green mesophyll matter will tell you its leaf. So try to understand the plant part and then go for if it is a bark, what are the possible drugs and then what are the possible features I should see in one particular type of bark. Say for example, cinnamon, I should see the volatile oil glands. If it is a kurchi bark, I should definitely see stone cells and so on. So it's cinchona versus cinnamon. So if you're seeing cinchona versus cinnamon, now if I have to see cinnamon, there are two varieties. If you clearly remember, there is cassia cinnamon and there is cinnamon zelanicum. So if I'm dealing with cinnamon zelanicum, my answer comes very simple because cinnamon zelanicum doesn't have a cork. If I am seeing cork in the powder, my cinnamon zelanicum is not there. Okay. Now coming to your cassia cinnamon. Now in terms of cassia cinnamon, there you see a cork and you will see a cork in your cinchona as well. So both of this bark are very similar. Now the differentiating feature of that are your phloem fibers. So maybe you will see your phloem fibers and oil glands. Okay. So in your cinnamon, you will see specifically your oil glands which are there. Now, when you see your oil glands, you know that it's an oil containing drug. So you can go for cinnamon, whereas in cinchona, you will see more of this stone cells which are there. And I think you just check with the phloem fibers as well. So does it answer you? So I'm sorry. It's just a small correction. I think Kurchi has stone cells. Your cinchona doesn't have stone cells. Your cinnamon has stone cells. So just a correction there. So, yeah, yeah. So you just check for the presence of oil glands, stone cells, and phloem fibers in your cinnamon, and only phloem fibers in your cinchona. The more better would be to do a quick TLC, but if you're seeing powders, that will not stand valid. So I'm just on a transverse section of cinchona, and the transverse section of cinchona shows a good medullary rays, secretory canals, as well as phloem parenchyma. So definitely you will see a lot of phloem fibers there, but you'll not see any stone cells in your cinchona. In cinnamon, just let me or let us put a cinnamon there. You definitely have your oil glands and your stone cell layer, which is very important there. So apart, if you see or if you want to differentiate between Cassia cinnamon and Cinnamon zelanicum, your Cinnamon zelanicum directly starts from the stone cell layer, whereas your Cassia cinnamon will have cork, it will have your parenchyma, and then it will have the stone cell layer. Just Okay, I'm not able to broadcast it, but you can just quickly Google in the uh, TS of cinnamon and a TS of cinchona, and you'll see the little difference between the two. So with this, we come to an end. So for your help, I'll just maybe put in some supplementary material if required and if possible on the portal. And you can feel free to put your queries in the forum. We'll be happy to address them. So maybe, the same queries you can put in the forum and we can have a small PDF as an attachment, which you can go through for your reference to some of these queries. So thank you everyone for your time and joining in the session. See you in the next live session. Till then, happy learning to all of you.
साथ होते आपण तुमचं पूर्ण 1 तास चाललं